So today in routine diagnostics of lymphoid malignancies, we are using FISH technology to detect certain genomic aberrations. It can be deletions or amplifications or certain translocations. So these are standardized assays with probes that detect these specific genomic aberrations, and we hybridize these probes to either smears, peripheral blood or bone marrow, or you can also use this probe on FFPE material. And this guide us, it can have diagnostic impact, but also prognostic impact for the patients. And besides doing FISH analysis, we have also been doing Sanger sequencing for a number of gene mutations, for instance, TP53 gene screening in CLL. Also, there can be sort of targeted hotspot mutation detection for certain mutations. But what has happened the last 10 years is the introduction of next generation sequencing or massive parallel sequencing. And with new instruments, sequencing instruments, we can now detect, or we can investigate higher number of genes simultaneously. So we can do targeted sequencing, or we call it gene panels, where we fish out the most interesting genes for a certain entity that we can then analyze at the same time. So, for instance, in CLL, we have shifted from Sanger sequencing to next generation sequencing of the TP53 gene. And in this way, many centers are not only investigating TP53, but also assess the mutation status of a number of other genes for research purposes. For lymphoid malignancies at large, there have been different gene panels developed, including genes that are currently mutated in different lymphoma entities. So these gene mutations could be correlated with, they can have diagnostic impact to refine the disease classification in lymphomas, but they can also have prognostic impact. So in Sweden, we have developed gene panel for lymphoid malignancies that include 252 genes that covers the most recurrent gene mutations in both B-cell and T-cell malignancies. And we are testing this right now, the final testing before clinical implementation. And we have a pilot study here at Karolinska where we are using this panel on patients with lymphomas to see what it adds to screen this large number of genes in each case. And we have molecular tumor boards in order to sit down together, the pathologist, the geneticist, the oncologist, to discuss what does it add to do this broad gene panel for the individual patients. So hopefully this will inform us how we can continue using targeted sequencing also in lymphomas. But today, I mean, we are using this NGS-based screening primarily for TP53 mutation screening in CLL, but also for some other hotspot mutations.